Okay, so a very good afternoon and welcome to this innovation workshop in collaboration with Thermo Fisher Scientific. So we're part of the Genesis Digital 2020 conference. So I'm very delighted to be welcoming you to our session today. And we're going to hear from three speakers who are going to share their expertise on a variety of chromatography and mass spec workflows. So just a few housekeeping rules before we start. So if you're not speaking, it would be great if you can keep yourself on mute and camera off. And then also just to highlight, we are recording this session and it will be available via the Genesis conference app up until the end of the conference on the 7th of Jan. So we also finally, we have the chat box enabled. So after each presentation, if there's time, then of course we can jump to that. So please don't hesitate to drop any questions in. And because we're a small audience, then, you know, we can unmute and have an interactive session. So without further ado, we're going to go into the first speaker, so Denis Calnan, who is going to talk to us today about HPLC LCMS methods that can be used to characterize the quality and safety of biopharmaceuticals. So over to you, Denis. Thank you.
Thank you, Alicia, and good afternoon, everybody. Yeah, I'm Denis Calnan. I'm the Business Development Manager for the pharmaceutical market for Thermo Fisher Scientific. This afternoon, I'd like to give you a little overview of the kind of techniques that we use to provide service to the biopharma industry, really, and particularly to look at the techniques that are available now to look at the safety and efficacy of modern day biopharmaceuticals. So for those of you that don't know Thermo Fisher Scientific, we're a large global organization, and our mission statement is to help our customers make the world a healthier, cleaner and safer place. And we do this by a value proposition of having a leadership in innovation and technology, and also a large global reach. And if you look at some of the brands that are under the Thermo Fisher Scientific umbrella, they really allow the industry to look at all aspects, so we can cover all bases really, and do this globally from all sides of analysis, from sample preparation through to the analysis of data, and then creating meaningful answers. And if you look at some of our business segments, we're very strong in the life science solutions, in research, particularly PCR. That's one of our major techniques. Also, providing laboratories with the necessary equipment and also storage, etc. We also have a specialty diagnostics division. But today I'm probably going to focus on our analytical instruments, and particularly along the lines of chromatography and mass spectrometry as two key techniques to analyze biomolecules. So I'm going to introduce some of the workflows, but my two colleagues later on will give you a more in-depth look at two particular cases, so two case studies. So if we think about biologics and biopharmaceuticals, here we've got a molecular model of a monoclonal antibody, 3D molecular model, compared to aspirin. And immediately you can see the physical size of these molecules are very different, and the complexity is vastly different. If you look at aspirin now on the right, it has a molecular mass of around 180 Daltons, so a fairly simple molecule that can be easily synthesized. The monoclonal antibody, on the other hand, is approximately 150,000 Daltons, and it's a complex glycoprotein, chains of amino acids and peptides linked together with a particular set of glycans attached. So there's a lot going on, and with that in mind, you therefore need to measure the quality attributes of this molecule in depth. So in the same way that aspirin needs to be manufactured in a quality environment to provide a quality product and a safe product and an efficacious product, It's the same for the biopharmaceutical industry. They have the same challenges. But of course, the complexity needs better analytics, so much more high-performing analytics. So why would you need to come to actually characterize these molecules? Well, as I say, being a complex protein, they are on a string of peptides and amino acids. They're open to modification during their production and also during their formulation and, of course, their storage. And certain molecules on the monoclonal antibody can be changed. And this change can either affect the actual efficacy of the drug, or worse still, it could have a detrimental effect on the patient by providing perhaps an immune response, or again, the molecule not working properly in the therapeutic setting that it's designed. So some of these characterization issues that we have are we'd look at the glycans on the molecule, we'd look for oxidation. We'd also look for aggregation, which is a common problem. So these are complex proteins, so they literally stick together in solution. This can then cause immune responses. So there are many different needs in characterization, and chromatography and mass spectrometry can provide solutions here. These are some of the systems. I won't dwell on it too much, but these are just some of the systems that we sell as part of workflows here. And importantly, the most important thing first is to actually prepare the sample well. So the sample could be a pure drug, or it may well be in a matrix of a biological fluid like plasma. We then need to separate out the components correctly. So for that, we use high-performance chromatographic systems. One of our key systems here is the Vanquish UHPLC chromatographic system. And then we'd need to detect the molecules. So we want to identify them and quantify them. So what have we actually got? Are there any differences compared to, say, standards? And how much of that have we actually got? So here we can use mass spectrometry, which actually measures the mass of a compound. And then when there's a chemical difference in that compound, you can see a difference in mass. And this can be interrogated in databases and data analysis. And with various forms of mass spectrometry, but the key mass spectrometry for Thermo Fisher Scientific is the Orbitrap, and particularly the new family of the Exploris Orbitrap family. Moving to the chromatography, I'd just like to highlight the UHPLC system that we use here, and you'll see this in the next presentations. So it's the Vanquish system. So this has been designed with robustness in mind. It's been designed for biocompatibility, ease of use, and easy servicing. So it complements the mass spectrometer in that you can combine them to work together within the same software and also in a compliant environment, a GMP environment. But all these analytical systems, it's all very well to have this high-performing analytical system, but the key thing here is workflow. So people, regardless of the industry they're in, they need answer to their complex questions and challenges. So the idea is that we've put together some unique workflows that are a one-stop shop, if you like. So bringing the power of the analytics together with a meaningful workflow, that you've got the sample prep, the dedicated analysis, the data analysis, and then giving you a meaningful report at the end of it. And that allows confidence in the lab. You can minimize errors and have compliance. So if we look at in depth at one or two of the workflows, so typically in a biopharma characterization scenario, you'd look at the intact protein itself. You just look at the protein as it is in the formulation. And that can tell you an awful lot of information. Looking at aggregates that we've already mentioned, but also looking to see are there any major mass differences. And this can be done by LCMS. You can then also take the molecule and break it up with enzymes and look at the individual components and really zoom in and look for specific modifications that might have happened through the production process or perhaps during the storage. And this is peptide analysis, so peptide mapping. We can also take this one step further with a method that we've produced in conjunction with some major pharmaceutical firms, which is the multi-attribute method. So this is a way of taking a single analysis, but being able to provide many different parameters in the one analysis in a compliant environment. HDX looks at the structure of the molecule and also the binding sites that the antibody may interact. And also, we can also look at large molecule bioanalysis. So in the same way that people need to look at the efficacy and the safety of a drug once it's in a patient, how it's broken down, how it's metabolized, and this is very common DMPK you may have heard of in the small molecule world, do this in the large molecule world as well. So we have the tools for large molecule bioanalysis. I wanted to mention one of our collaborators. So while we're making these workflows, the important thing is to keep them industry relevant. Keep the idea that, you know, these are relevant to the working laboratory. And we've teamed up with, for a number of years now, with an institute in Dublin in Ireland, the National Institute for Bioprocessing Research and Training, or NIBRT for short. And here, this slide really, it's just to illustrate the breadth of collaboration we have here, that they've created the workflows for us. They try and test them. They test them to the nth degree to show that they're robust. And there are a particular website here that I could point you to, which is thermofisher.com/NIBRT, N-I-B-R-T. Here you'll find a wealth of small videos, information, all the different profiles and all the different characterization workflows I've spoken about. So if we look at a particular workflow here, which is for aggregates, so remember I talked about the idea that the antibody is a very complex glycoprotein and often sticks together in solution. Now that's bad. You don't want that. You want to engineer the molecule in such a way that this doesn't happen. But in order to monitor this, we have a particular workflow here whereby we use size exclusion chromatography, the MabPack columns, together with the Vanquish UHPLC system, and we can link that either to a UV detector, or we can actually link it to one of our Exploris mass spectrometers. This is also all connected to the Chromeleon data system. So this will acquire the data, process it, and give you a meaningful report. If you look at some of the data here, this was a study that we carried out looking at lifetime stability of the columns. So often when people design a method, they want it to be robust, and they want it to be easy to use, and also be cost effective. So the size exclusion columns, they're reasonably expensive in the real world, to be honest, and it's something that people are often concerned about. How long will my column last? How many injections will I get from it? How robust will the data be? Well, here we've done a study whereby we've injected a monoclonal antibody into the analytical system and looked for consistency over time. So on the chromatogram on the top left there, you can see that you've got the main peak of interest, which is your drug, and then you have some of the aggregates that are appearing before it. And then when you look at the zoom in, there are some fragments, smaller fragments. So ideally, you want most of your product to be in that main peak. But the interest part of this study is that NIBRT did this for us. Long-term study, they injected nearly 2,000 injections simultaneously and continuously. And the performance of the column was maintained and the quality of the data was maintained. So this provides a really good cost-effective method. You can run these columns for a long time. And this is a particular feature that is within the Vanquish system. It's to do with the injector that allows and preserves the column lifetime. So if we look at the data that we're looking at as well, so this is completely controlled by Chromeleon in a compliance-ready environment. So this is the software. It's quite unique in the chromatographic data systems that it can control both the chromatogram, the HPLC, and the mass spectrometer. It can do our whole Orbitrap platform. All of our instruments are mass spectrometry instruments. And it has particularly nice features in the biopharma world that it can look at the intact protein convolution. It also has compliance-ready reporting. So there's audit trails in there, there's electronic signatures, and all the things you'd expect in a GMP laboratory in one place. We can also take full methods and bring them together in a full processing method called an e-workflow. And Chromeleon 7.3 is our latest version of this. And again, there's lots of information on our website. So just to summarize, we help you succeed in the pharma and biopharma applications from early R&D right through to development into a routine environment. And Thermo Fisher Scientific has the breadth and the knowledge and the depth of instrumentation, the software, the sample prep that will allow your lab to work in this environment. We've many applications that are relevant to the biopharma industry, particularly intact mass, peptide analysis, HDX, and then the large molecule DMPK that I spoke about. But more importantly, we've a very broad service and support organization so that we can support these workflows continually and also and help people improve the production and the efficiency in their labs. Thank you for your attention. If there are any questions, I'll answer them, but my email address is also on screen and we can be easily contacted. So thank you for your attention. Thank you.
Thank you, Denis. Thanks very much for setting the scene with that presentation and some of the different methods and techniques. Just a question from me, and it's perhaps quite an obvious one, but when you say UHPLC, is that high performance or high pressure? Are the two interchangeable?
They are probably interchangeable. I think the common language now is performance. People talk about performance because it's ultra performance liquid chromatography, so UHPLC.
Perfect, thank you. So if there are any other questions, you know, we've got some time, just pop them into the chat box and we can get back to those at the end of the session. But following on from that presentation quite nicely, we are now going to hear from Mindu, who's going to talk about applying UHPLC MS to identify the key metabolites in cell growth culture media as a way of improving growth and yields. So Min, I can see you started sharing, wonderful. Over to you, thank you.
Thank you, Alicia. I'm trying to share my slide. And
that looks good, yep.
Okay, you can hear me well, right?
Very well, thank you.
Okay. Hi, everyone. Good afternoon. My name is Mindu, and I am the Biopharma Application Team Manager here at Thermo Fisher Scientific, Chromatography and Mass Spectrometry Division. Like Alicia said, today I will discuss with you how to use the state-of-art LCMS techniques to profile the key metabolites for cell culture media analysis, which is very important for the biopharmaceutical development. So if you are familiar with the bioprocess during the biopharmaceutical manufacturing, you may often hear about spent media analysis, which refer to the analysis of amino acids and nutrients that the cells use up during their growth cycle, and the metabolites that are produced by, as a byproduct by the cells. All the analyzed are present in the supernatant, and which include amino acids, nutrients come from the cell culture media and the dairy phase, as well as the nutrients which can be consumed, which can be monitored as well, and the byproduct that's secreted in the supernatant. In the biotechnology industry, where there is a need to sustain a high cell density and increase cell cellular productivity, the development and optimization of cell culture media is very crucial. Thus, the accurate metabolite analysis is very critical for the developers to really understand both the intracellular and extracellular environment in cell culture, which could lead to a greater understanding of the bioprocess, which can impact the productivity and the product quality. During the cell growth, the increase or decrease of those metabolites can have significant impact about the titer and the CQAs of recombinant therapeutic products. On the right-hand side, which listed some of the, how the industry uses the cell culture media analysis to help them to understand the bioprocess. Our customer wanted to develop a quantitative assay which can monitor and quantify multiple classes of cell culture media metabolites simultaneously in a single LCMS analysis run. The developed quantitative assay needed to have the high throughput and the robustness required to support the upstream process optimization and eventually the quality control. However, to develop such an assay can be very challenging due to the diversified chemical and the physical properties of different classes of analytes, such as the amino acid, vitamin, and the sugar you see over here. They are very dramatically different in terms of their structures. And many of the metabolites are also present in wide concentration range, can be up to 5 orders of dynamic range required to analyze those compounds. Here we talk about UHPLC-HRAM MS solution developed in the group for cell culture media metabolite analysis. And as Denis showed in the previous presentation, we utilize the combination of the UHPLC as well as mass spec for this analysis. And in this method I show later on, we use the Thermo Fisher Scientific one-click Horizon UHPLC connected to the Orbitrap Explorer's 120 mass spectrometer. And in terms of data analysis, we use the Chromeleon data system software. And in this solution, we can simultaneously analyze multiple class of metabolites as required by our customer in a single LCMS run with a very fast positive and negative polarity switch. Since all of the metabolites are very structurally different, they can either carry the positive or negative charge. So it's very important to analyze them in a single run on the fly. And with the newly introduced Orbitrap Explorers series mass spectrometer, they are designed to be easy to use as well as easy to set up and maintain. This mass spectrometer also features faster scan speed, which will enable enough data point across a peak, a chromatographic peak for precise quantification. It also features very high mass resolution up to 120,000. And easy IC internal calibration feature enables excellent sensitivity and sub-ppm mass accuracy, which is very important when you analyze those metabolites in a complex matrix. So as Denis alluded before, the single compliant ready software, Chromeleon CDS, will enable our customer to control our instrument as well as process data while maintain the data integrity required. So with this method, we rely on the high resolution accurate mass spectrometry data, which have really high confidence of the data quality. In addition to that, not only it can perform quantified targeted analysis for those metabolites. When you have an unknown metabolite show up, it can also identify those unknown metabolites. It has a flexibility also doing additional time TSIM as well as a TEM-IMS2 for challenging metabolite analysis for improved sensitivity as well as more structured information. In this slide, we show that in this method we developed is for 96 cell culture media metabolites. They're very diverse, including 35 amino acids, 20 nucleic acids, and 8 metabolites involved in TCA cycle, 15 vitamins, 6 carbohydrates, and some other metabolites shown here. The first step we want to develop on this method is excellent separation, since we are targetedly quantify those over 90 metabolites in one run. We want a very good and robust separation of those metabolites to enable really great identification and quantification. Here shows the chromatographic profile of the across different amino acids. As you can see, they are well separated and the peak shape are really good. So this method is developed as a high throughput assay, so it's only 20 minutes total run time. And the next slide shows the representative targeted vitamin standards on chromatogram. As similar as the amino acid, you can see a great separation. The same also shows for the nucleic acid standards here. In addition to the high resolution separation, as I mentioned before, we operate this method with a high resolution. In this method, we operate at a 60K resolution for both positive and charged negative ion detection, and with the polarity switching. So as you can see, the zooming of the histidine peak over here, across the peak, we have a 14 to 15 data points. And if you can pay attention to the mass accuracy, so it's a 0 ppm mass accuracy is a spot-on detection of histidine metabolite here. And every data point, you actually see exactly the same mass detected, MOV detected. So this really enabled a very confident identification and the quantification of each metabolites. To demonstrate the method sensitivity and the dynamic range, we did an experiment which we mix 85 metabolite standard and diluted with water to make a mixture solution of the final concentration range of 500 nanogram per ml to 1000 nanogram per ml per metabolite to mimic the complex matrix effect. And then we use the Thermo Scientific Pierce amino acid standard, which is a standard of a mixture of 18 amino acids supplied at 2.5 micromole per ml concentration. And then we spike that into the mimicked metabolite matrix at the following concentration from 250 nanomole per ml all the way to 0.025 nanomole per ml. With that, we can demonstrate we can have very robust quantification across the 3 replicates. As you can see, the CV is very tight across this concentration range of 4 order of linear dynamic range. So it's a very sensitive assay as well as cover a wide dynamic range. So that has been established for all the 18 amino acids. We only show one representative amino acid over here for the leucine. The next case study, what we really want to see is how we can apply this method for the metabolite analysis for the cell culture media. In this case, we use the 3 MEM samples were diluted with 0.1% formic acid in water to make the final dilution series samples in 10-fold, 50-fold, and 100-fold dilutions respectively. So we can demonstrate that with the dilution, you can see the trend of the metabolite reduced. And one of the internal standard was used is the 2-isopropyl acid was spiked in each of the dilution series samples as a final concentration of 1,000 nanogram per ml for the relative quantitation can be taking place. And as you can see, all the three cell culture media is showing in the left-hand side. And on the right-hand side, on the top, you can see the measured peak areas of the glucose from the 3 replicates in the highest concentration with a 1 to 10-fold dilution all the way tracked down to the 100 dilutions. You can see very reproduced result across three replicates in all the concentration level, as well as a consistent reduction of the peak areas here. And two of the lowest concentrations of metabolites are showing here at their lowest detection level in this experiment to reach 1 nanogram per ml for biotin, as well as riboflavin. And both of them show very good peak shape as well as very good signal to noise. So across the three cell culture media samples, we then showing here is you can easily trend the metabolite levels. So as you can see the right-hand side, and we show different metabolite classes over here. And the blue line represents the MEM alpha media, and the red line represents the F12, and the green line showing the MEM or the basic media. And you can see the clear trend. First is a very different concentrations across the 3 different media type, as well as you can see a consistent trend when we showing up with the dilutions series. So the Chromium software was used for both the data processing and the visualization over here. And the metabolite changes over the different samples can be easily monitored and visualized. And with the visualization on the y-axis, we are using the relative quantification of the peak areas of a target versus the internal standard peak area. So the sensitivity, as what I showed before, can go down to the 1 nanogram per ml was observed for this particular experiment. So what we showed in the previous is a targeted analysis. When we run the full scan LCMS analysis, then we have a target list to isolate those metabolites and to show their quantification levels. But this method can also go beyond the targeted cell culture metabolite profiling. If you have an unknown metabolite, then the full scan MS can give you additional information to go retrospectively looking for those unknown metabolites. In many cases, in the real life scenario, you will need that capability. For example, if the titer of different bioprocess was dramatically different, but from the existing metabolite profiling studies, you cannot draw the conclusion. So you might want to look into the untargeted cell culture metabolite analysis and find if there's additional differential metabolites showing up, can really you can investigate and try to understand what caused the issue. So for the previous experiment that I showed for the media analysis, here we show that is the MEM alpha media. And in this list of the media component, you can see everything with the blue texts are the metabolites already included in the targeted analysis. And versus, let me see my mouse. These 2 particular metabolites we highlighted in the red boxes. And these two metabolites are not included in the initial method when we developed that method. So for this, we can go retrospectively looking at our data. Since we have the high resolution accurate mass data, we can really, using the molecular weight information we know about this metabolite, and went back to retrospectively to extract chromatographic. So as you can see clearly, you can see from the 10 times, 20 times, 100 times dilutions, you can see this extracted ion chromatogram shows reduced peak areas of this particular metabolites. And with the 3 rounds, same as before, it's very good reproducibility. The percentage RSD is less than 5%. And you can see the trend of this metabolites go down with the concentration represented that by the peak areas. In addition to using the accurate mass information, if you do need to have additional information about the structure information to boost the confidence of that identification, this instrument can also offer the capability to run the tandem mass spectrometry experiment. So you can isolate the parent ion and fragment it in the to achieve the MS2 data. So in this case, you can see the parent ion and signature fragment. So that really can help you to provide structure elucidation information and confirm the identification of this metabolite. Here at the end, I want to summarize. So this is, we show here is a platform HRAM full scan MS method that has been developed for cell culture media metabolite profiling, which is easy to set up by using online switching full scan positive and negative MS. The faster scan speed of the Orbitrap Explorers 120 mass spectrometer really can enable our customer to do this analysis, provide enough data point, scan points, and excellent mass accuracy for very accurate and precise quantification and identification of those targeted metabolite compounds. The comparable sensitivity and the linear dynamic range was established versus some of the other methods was developed on the triple quadruple mass spectrometers. And in addition to this targeted analysis of those 96 metabolite compounds we show here, the unique advantage of this platform method is to provide the quantitative and the qualitative analysis of additional compounds beyond the initial target list set up by the method by retrospectively looking at the HRAM mass spectrometers for further data analysis. At the end, the Chrominian CDS software was used for data aggregation, data processing, and reporting, which can be deployed in both regulated and non-regulated environment to facilitate the method deployment and the transfer. With that, thank you all for your attention. And if there's questions, I can answer that as well.
Thank you very much indeed, Min, for that very interesting case study focus there on such a powerful tool, you know, highlighting the sort of diverse range of compounds you can do in one run. And also, you know, the fast efficiency must be really, really helpful for customers indeed. In the interest of time, I think we'll move on to the next presentation, but the chat box is enabled, so if anyone does want to drop any comments or questions, please do. So I'll now hand to Ken Cook, who's going to cover advanced methods for analysing compounds that have applications in cell and gene and vaccine manufacturing. So over to you, Ken. Thank you.
Hi there, can you all hear me quite well?
Yes, we can. Thanks, Ken.
That's great. We're going to look at the power of chromatography here And
Sorry, Ken, we can't see your camera, but if you don't have to have it on if you don't want to.
yeah, I think I started to share the screen before the
No,
camera.
oh, no problem. No problem. That's
Yeah.
okay. Yep. You can just go back to your slides.
Yeah, so we'll go from there.
Yes, no
so
problem.
what we're going to look at here is the power of chromatography. Now, Denis and Min's both given a good explanation of how you can run methods and what the equipment looks like. Now I'm going to give you an idea of some of the applications we can actually use for that. And if you can see the applications we've got here, we've got the biopharmaceutical workflows in general, where you've got a series of different applications you can use in biopharmaceuticals, quite often monoclonal antibodies, but not entirely restricted to monoclonal antibodies. Denis has already talked about the aggregate analysis. Now for aggregates, it's an aggregate's bigger than the single monomer molecule, so it's a size separation. So they're using size exclusion as a column, and you can use HILIC as well, but that's also available with UV detection. We can put it on with an MS to get accurate mass, and we've also got Mabs, which you can use as well to give you an idea of the molecular weight. If you go to charge variant analysis, it's separating molecules by charge, and even just small molecules small changes can affect the change in charge that we can separate on an ion exchange column. So here we use an ion exchange, but again, we can use pH or salt gradients to look from there, and we can use UV, and again, we can connect this to high resolution mass spec. Now, when you want to do protein analysis in the intact form, we can use HILIC, which is a salt-based method, which uses the hydrophobicity but keeps the molecule in the native form. If you want to denature it, we can go into reverse phase. Now, the column we'd use for reverse phase here, because you've got an intact protein which is quite big, we'd use a column with quite a wide pore size. Now, if you move across to peptide mapping, we're also on reverse phase, but we're going to choose the column differently because the peptide's now a lot smaller than the intact protein, so the pore size you use on this column will be a lot smaller. And again, you can use UV or MS with any of these techniques. For glycans, we tend to release the glycans, and glycans don't bind well to ion exchange or reverse phase. In this case, we're going to use a HILIC column or mixed mode columns here to separate the glycans out. And we'll show you a bit more of that later on. But again, that can be used with fluorescence on a standard HPLC or mass spec and even CAD. Now, we're also going to look at oligonucleotide analysis. And again, a totally separate column for oligonucleotide analysis, one that's designed for oligonucleotides, but you can do that again by ion exchange and reverse phase. And the columns can be made mass spec compatible for this as well. And we'll look at bioanalysis and even where you've got the media, which Min showed before. So if we move on, you can see the range of columns you need. Now, to do bioanalysis, it's not just a matter of having an HPLC on the front, binding it to a mass spec and looking at data analysis. There's quite an arsenal of chromatography columns you need to do biopharma. And Thermo Fisher Scientific have got quite a good array of these. And what you can see, they're named usually after the application. Mab packs are usually designed for monoclonal antibodies, but as they're proteins, you can use them for most protein work. And again, the Pro pack designed for proteins, but because monoclonal antibodies are protein, you can use them for monoclonal antibodies as well. And we've got DNA packs, which again, for any oligonucleotide, including RNA, and the messenger RNAs we're now using in the vaccines. And of course, we've got hydrophobic chromatography in glycan pack columns as well. So it's a whole array of these columns. Now I'm going to go and show you what the columns do. But as I've said, the machines need the columns, but the columns actually need the right machines as well. Now, when you're looking at ion exchange chromatography, you're looking at change in charge, looking at changes in charge. And some of these variants can be something like you've got 150,000 Dalton protein in a monoclonal antibody and an extra lysine put on there, one amino acid with one charge, and you can separate that quite efficiently on an ion exchange column. So that's really a powerful separation. Deamidation is another one which pops up here in the acidic variants. And that's just taking the deamidation site off a protein. And again, you've lessened the charge in terms of a positive charge, so it turns up as an acidic variant. Now, there's various columns you can get for this. Usually we'll look at strong exchanges and weak exchanges, and they've just got different groups on the front. But in general, we're using follicular resins, which means it's a solid base. You've got a hydrophilic coating on the top to stop hydrophobic interactions, and your charges are presented on the end of the polymer chain. So you've got interactions at the top, and what interaction you have at the end will depend on what functional group you use. Sulfonic acids and carboxylic acids are used for cations because positive charges like to attract the negative charges. If you use a positively charged protein or analyte, you'd have a negative charge on your protein, but the other way around, you'd have a negative charge on the if you've got a negative charge on your analyte, you'd want a positive or an amine as a functional group on the protein. Now, some of these separations are salt-based, and not just salt-based, they're corrosive salt. It's usually chlorides or halides. So you need a machine that's not going to rust from the inside out. And when we've got the Vanquish system, it's totally not inert. So it's not going to rust, and it's not going to bind to your proteins. And the tubing we use, the finger-tight tubings, but the material's NP35N alloy, which is the same material we use in surgical implants. So that's not going to rust either. So in this case, the whole system's inert. It's not just the flow path, the whole system's inert. So that pump is going to last for a long time at high salts. But after saying that, that pump's also got to pump solvents. So you're asking a lot of an HPLC system, and the Vanquish system's very good at that. Now, when you look at ion exchange, we've made some buffers to make life a little bit easier and run pH gradients. These are called CX1 gradient buffers, and the buffers that are inside there are strong Zwitter ionic buffers. We're using Zwitter ionic buffers because they haven't got a particular charge, they're neutral. So, and they cover a high pH range, and they're in there at a good concentration to give you a powerful linear pH gradient when you want to do charge variance. And the pH gradients, you're just gonna, for a monoclonal antibody, you'll bind in low pH. As you increase the pH, the protein will actually change its charge till it eventually becomes neutral, then just falls off the column. Now, if you try using different buffers, for example, if you use amine containing buffers, they'll bind to the resin. If you use an amine that's got two double charges on there, it'll bind quite strongly and difficult to get off. Now, we're using buffers that don't bind to the column, but they can go to the surface of the column and interact with the surface of the column. If you've got buffers which are negatively charged, they're not going to go anywhere near that resin surface. So here you've got the ability of having buffers you can't get off the resin and buffers that don't go anywhere near the resin. So the actual pH control at the surface of the resin where everything's happening can be quite erratic and different from the pH that's actually in the solution itself going through the column. So that's why we're using Zwitter ionic buffers. But to give you an example of how fast we can do this, normally charge variant analysis was designed as an hour-long method, 60 minutes. And it was said it's not a high throughput method. That's because they were using salt gradients with long columns. With a pH gradient, you can use short columns. And this is in a case of a 10-minute scouting run. And if you look at the ones on the right here, once we've done the scouting run, we know where it's coming off. We can optimize the gradient around the pH where the analytes are coming off. And here's examples of 2-minute runs, 2.5-minute runs. And so you've got the speed, but if you look at the resolution, that resolution is just as good as what you'd get in 60 minutes with the salt gradients. But now you've done it in under 2.5 minutes. And what you've got there is a very, very stable method. And you can do the method development in an afternoon. So it's quick to develop the method, it's stable, and it's high throughput now. Now, we can also direct inject this through into MS. Now, the problem when you've got with the CX1 buffers and salt is that you can't put salt and charged things into a mass spec. It's got to be volatile. So we've changed the buffer system to a volatile buffer system. Now we've developed this, we've published this, and the publication's on the bottom of the screen there. And if you use that buffer, it works very well with the short column. But we're not sacrificing resolution here. That's a very, very good separation. If you look at peak 1, that's really going through the roof. We're looking at things that are very low, so we've got the sensitivity. And if you look at the connection into MS, we can now see those two peaks are 128 Daltons apart. that's equivalent of a lysine. So we know that's lysine truncation. You've got another set of peaks there that are 128 Daltons apart. We know it's lysine truncation, but it's actually got an additional succinimide on there because it's got an extra -18 on there. So what you're looking at there is two modifications in one go. The mass spec seeing that quite easily and identifying it. And is it reproducible? Yes, if you look at the three marked in green with the green arrows, they're deamidation peaks. And that's 1 Dalton on 150,000 Dalton protein. And the resolution of the MS and the technique is good enough to see that. And it can see that reliably. And you can see you've got 3 injections here of the first peak, the second and the third marked in green there. And we're seeing that deamidation variability quite easily. You're also picking up glycans here on the bottom right, and the glycan distribution, you can get that from every single variant. And it's a very powerful technique. Now that's cations, but we can do this with anions as well. We've developed a buffer that's volatile, and it'll actually pick out the anionic proteins with an anion exchanger rather than the cation exchanger. And here you've got the difference where it's now picking up negatively charged things. So if you've got a phosphate group, which albumin does have, the one at the very, very start, a low level amount of, doesn't have any phosphorylation. Then you've got a single phosphorylation as you're moving in, and the peak highlighted in yellow here is 2 phosphate groups. And if you move even further to the right, you're starting to see salic acid groups or charged sugars appearing and separated on column. And again, each of these peaks you can pick out the glycans from. And we've now picked up 150 isoforms of albumin, which is about 3 times more than anyone else has ever done. Now, I'll move on to some of the other columns. Here's the example of hydrophobic interaction. And again, completely different mechanism. We're forcing things onto a column with hydrophobic interaction. And because we're looking at hydrophobicity, it works very well for conjugated monoclonal antibodies that are being conjugated with a drug. So you're looking at antibody drug conjugates here, and we can easily pick out the drug to antibody ratio just from the peak areas in the separation here. And that's done very, very quickly on a HILIC column. It's also very commonly used to look at oxidation variants as well, because the oxidation variants often cause a conformational change, which will change the hydrophobicity of the protein. So they're good columns for those. Here we're looking at glycans. And again, it's a very reliable glycan separation. And it's a different mechanism. Again, it's HILIC, it's hydrophilic interaction chromatography rather than hydrophobic. And because the glycans, we can force those on because they are a hydrophilic molecule with those OH groups. And you've got a nice separation there. And the column we're using here is depicted here graphically as it's a superficially porous resin. It's a solid core, and the resin's on the outside of that solid core. Now, in addition to that, with the glycans, some glycans can have salic acids on them, and that enables a charge. So we can now start to separate by charge as well. Whereas the HILIC column on the right here, which we showed you earlier, one separate by charge, sometimes they have charged variants going up on top of each other. It's not its major form of separation. Here we've got a mixed mode column now where we've got reverse phase interactions and ion exchange interactions at the same time, or the HILIC column with ion exchange. And again, we're now starting to separate the glycans by conformation, but also into groups of charge. You'll get the neutrals in one group, monosylated, di, tri, tetra, all the way up. So now you've got a different separation, again, depending on the analyte. And can we use these mixed mode columns for anything else? Well, the pharmaceuticals produce drugs, but those drugs have counter ions. And here's an example of penicillin with its counter ion, which is potassium. Now, potassium's not UV active. Penicillin, you'll get some sort of UV reaction, but here we're using a CAD detector. Now here we're actually charging up the molecules to allow it to be a universal detector. It more or less picks everything up. So where you've got the potassium, here's the potassium picking up, and it's binding to the column because it's also got an anion exchange site on the column. And it binds the penicillin because of the reverse phase and a little bit of the ion exchange as well. And this is another drug on the right with malate as a counter ion. So you're picking up both of these analytes and the small molecule drugs at the same time using a specific detector in a specific column. And this is an idea of these C18 characteristics. I often see people, oh, C18 columns are C18. It's actually not. And when you look at the diagrams here, we've got various C18 columns. We've got an AccuCore here, another AccuCore, which is designed for MS, another one which has got an embedded group in there. You've got a C30 column, which is a longer chain. And you can see all of those four that I've mentioned have got similar selectivities, but they're actually quite different. And if you look at the spider plot, we're looking at ion exchange interactions, which is IEX. We're looking at shape selectivity, which is the SS, the hydrophobic retention, which is the HR at the top. And you can see same type of column, but the selectivity will be slightly different. And again, if you've got different types of molecules, you might want a different functional group on there, such as a biphenyl group, which has got a lot of ion exchange interaction on it. And that gives you different selectivity again, and again with the phenyl X, which has got a lot of shape selectivity. So you pick the column for the type of molecule you want to separate. And again, with just peptides, you'd look, peptides are fairly big. They're not as big as protein, but you'd still want a good pore size on there. So you want at least 120, 150 angstrom pore size. And the longer your column, the better your separation. So here we've got a very long column, which is allowing us to separate the amidation sites on a peptide. Now, it's a very small variation on the peptide for basically a reverse phase column to pick up, but the column length allows you to do this very, very well. And you can see another C18 column on the left, which is a smaller particle size, which creates a big back pressure and prevents us getting this longer column length. It just doesn't do that separation as well. So even with the C18 world, you really need to pick the column for the separation. And I'm going to move on now on to how we can do oligonucleotides. And again, we've got two columns for this. It's a reverse phase column and an ion exchange column. But there's a separation shown here on the reverse phase column, but because we can put that into a mass spec, we've now got the mass spec on the end at high resolution, and we can do deconvolution of the intact molecule, or we can do fragmentation of the molecules and actually sequence it and annotate that sequence automatically in the software, which is the first software to be able to do that. So you've got now a full way of, and a full workflow all the way from the sample column, the chromatography, the mass spec, and the data handling to allow you to do really good oligonucleotide separations. And that's now becoming quite powerful in the short interferon RNA therapeutics, the messenger RNA vaccines that are quite popular at the minute, and in gene therapy. So these columns and the workflow itself is becoming very, very useful. And what happens when one separation doesn't quite give you all the information you really need? And that's where we move into two-dimensional chromatography. And here we're using a multi-hard cut technique where we know the peaks that we can't separate, and we've got things underneath. Now we want to collect those peaks on a trap or a loop. I do prefer trap collection. And here we've got that peak there at the start. It's got two peaks inside it. We've got other ones all the way through where, in this case, we've trapped 4 different segments and 4 different peaks, and then put them onto a different column with a different selectivity to separate those things that didn't separate in the first dimension. And each of those separations you see highlighted there from the different peaks, they've come out quite happily and separated the additional thing that didn't separate in the first dimension. And that's a good separation to do. We've got that worked out pretty easily and pretty easy to use with the Vanquish 2D system. And again, here's another example of when you've got a peak which is very, very small, and it's difficult to pick up and identify. But what we've done here is that peak there is a small peak. The mass spec in this case, it's got enough sensitivity to see that peak. But if you look at the charge envelope you've got just underneath, that's a good, nice, sensitive charge envelope, even though the peak's quite small. And that is the raw mass data from one charge state. And if you look at the deconvolution underneath that one on the right, looks the same. So we're deconvoluting, getting good, accurate mass data. And you can see on the slide at the top left here, the top trace is the first dimension run. The second trace is the second dimension run done with volatile eluents. The first dimension had high salts in, so we couldn't interface that directly with mass spec. We've trapped them and then desalted with a volatile eluent, and the bottom trace shows you That area marked in a pink profile, we've got a pressure dip there. That pressure dip refers directly to the peat we've collected. Now, if we want to collect a really dilute peat, we can just do several runs and collect them onto a trap. And then we've got enough on the trap, we then do the full run and put it into mass spec. So it's a very powerful technique, even for small level molecules. So really, it's just to give you an example of, there's a lot of tricks you can do with chromatography, choosing your right columns, and you can even join columns together in more than one dimension and make sure you can get them into the MS, which is giving you that additional information you can't get with just UV on its own. So I'll open up that for questions now.
Thank you very much, Ken, for that highly technical presentation. I think what you've really highlighted there is just how complex these techniques are. So you really do need to know what you're doing. You really do need the expertise. You know, you've got the columns, you've got the buffers, you've got the methods, just so many different variables. So I guess that's where an expertise from Thermo Fisher Scientific really does come into play here to advise on all those different elements. So we are up to time, but I just want to highlight that Ashley Box from Thermo Fisher is on the Genesis app, so please do follow up and if you want any further additional information. But thank you very much to Thermo Fisher Scientific today for sharing your expertise on this very powerful technique, which, as you've just demonstrated, has so many applications for which are helping us to deliver new therapeutics. So with that, I'll close the session and just say, wish you all a good day, and just also to mention that our innovation workshops will continue next week on Monday and Tuesday, and then from the 16th of December, all of the content, including this session, will be available via the app. So thank you once again for your time. Take care.
Thank you.
Bye then.